Okay, so we left off last time just talking about how the cytoskeleton, if you will, we talked about it, how does the spindle change throughout mitosis, but it's all the cytoskeleton of the cell. And so somebody summarize this for me. What are the major changes in the cytoskeleton during mitosis or during M phase? Remember, there are two different cytoskeletal components involved. There are the microtubules, and they go from doing what to doing what during M phase? Yeah, Levi.
Holding the organelles in place.
Yeah, they go from holding the organelles in place,
Stretching things out.
yeah, to stretching the cell out, to physically grabbing hold of the sister chromatids and pulling them apart from one another. And so our microtubules completely reorganize. And they reorganize around what we call microtubule organization centers, or in this case, is the centrosome. Okay, the centrosome with its two centrioles. But our microfilaments also reorganize during M phase. Right? What do the microfilaments do during M phase? Yeah. Yeah, they form a contractile ring around where they determine the center is. Now, most of the time, and you saw this in lab, now all of you have been through lab, you should have found some cells that were in metaphase. For most cells, the middle is going to also be the geometric middle, right? It's like right in the middle of the cell. But sometimes you need to separate the cytoplasm unevenly, right? After the division, sometimes you need a big cell and a little cell. You don't see that a lot during mitosis, but you do see it happen a lot during meiosis, which we'll talk about next week. All right. I think it's pretty good. Yeah, Chris.
Just a quick side question. What happens for cells that are asymmetrical? How do they divide fairly evenly, or?
You mean cells that aren't typically spherical?
Yeah.
Yeah, I mean, so you'll see this especially happen, say, in the underlying layers of your skin, where you've got some cells that are more cuboid than they are spherical. But yeah, they'll typically divide along the same. And actually, during mitosis, they'll actually become a little bit more rounded as you start to stretch the cell apart. Yeah, and then plant cells, I mean, they'll stay cuboid. And then, but there you're really more concerned with building up a wall in between what's going to become the two cells. Yeah, Allison.
So do they have to make sure there's an even distribution of organelles, or do they just develop new organelles after they split?
It's a good question, and the answer is both. That they try to ensure an even distribution of organelles, but you also have some reestablishment that can happen following. Because some of the organelles there's only one of. Right, like the endoplasmic reticulum, both the smooth and the rough, the Golgi apparatus, there's really only one of those, the contractile vacuole in plant cells. And so those need to be replicated as well and then distributed evenly. But they can also be rebuilt afterwards. You know, the endoplasmic reticulum, it's membrane. The Golgi apparatus, it's membrane filling fluid-filled sacs. Those can be rebuilt. Yeah, that's a good question. It's both. All right, so the next question is this. How do the chromosomes move along the spindle? And again, what is the spindle? What makes up the spindle? Microtubules. And how do things move along microtubules during G1 when the cell's just doing normal cell stuff? Motor proteins. So how do you imagine we're going to answer this question? Motor proteins. Okay, so just to put this in context, we're gonna focus in on these kinetochore microtubules for a moment, although we'll also talk about the non-kinetochore microtubules as well. So here's a kinetochore microtubule. We call it that because it's physically attached to what? The kinetochore, which grows at the centromere of these chromosomes. And so something that was interesting is that the idea was that really all that's going on when these sister chromatids are pulled apart is that these microtubules are shortening. You're just starting to shorten them. But what's interesting is microtubules only grow and only shorten at one end. They only grow and they only shorten at one end, and it's called the positive end, which happens to be the end that's attached to the chromosome. And so because we knew this about microtubules, we're like, I bet that they're not shortening at this end because microtubules only lengthen and shorten at the positive end, which is actually over here. So then we're like, well, that's very strange that they're shortening at the end that's physically attached to the chromosome. Unless the microtubule is not what's physically attached to the chromosome, but rather there's something else that's attached to both the microtubule and the chromosome. But we'll come back to that in just a moment. So what they did was they basically bleached the region of the microtubules right next to the pole, and then they watched this bleached portion during mitosis. And this bleached portion stayed the same distance away from the pole throughout the entire process, confirming that the microtubules were not shortening at this end by the pole, that they're actually shortening at this end by the chromosome. Okay? Because this bleached portion does not get any closer to the pole until the whole thing is dissolved during telophase, where you dissolve the entire spindle and you reassemble the normal cellular cytoskeleton. Okay? Does that make sense? Okay, so they already suspected that the chromosomes must be shortening here because that's how microtubules work. They only lengthen and shorten at the plus end, and they confirmed it by bleaching a section of these microtubules and demonstrating that that bleached section stays the same distance away from the pole the entire process. So the chromosomes are shortened at the end. And the only way this can work, again, is if it's not the microtubule that's attached to the chromosome, because how can the place that's attached be shortening? Or if there's something attached to both. And again, how do things move along microtubules? Motor proteins. So what do you think it is that's attached to both the kinetochore microtubule and to the chromosome? It's a motor protein. And so they found them. So you've got what's going on here is you've got these motor proteins, kinesin, which is the same motor protein that walks vesicles along microtubules, is physically attached to the kinetochore and then walks along these microtubules. But it doesn't walk along them right on the end. There's a little bit of space where it could be shortening. And it basically shortens at the same rate that kinesin is walking it towards the pole. In the same way, kinesin walks vesicles either towards the plasma membrane or away from the plasma membrane towards the Golgi apparatus. Do you remember talking about that when we talked about the endomembrane system? Okay. Well, if you don't, remember it, because I know that was covered on exam 1, which won't be covered again on exam 2, but this will be, right? And an understanding of how this works will be helpful. Okay, so the motor protein, bless you, kinesin, is physically attached to the kinetochore and is walking along a shortening microtubule. And you see the microtubule is shortening at this end, right where the chromosome is, at basically at the same rate that kinesin is walking those chromosomes towards the poles. Okay, so to summarize all this, how do the chromosomes, motor proteins at the poles are pulling these kinetochore microtubules. Motor proteins near the kinetochore are walking the chromosome along the microtubule. But then we also have motor proteins working on those non-kinetochore microtubules, walking them in opposite directions, lengthening the cell. Okay? Similar to what's happening when you walk microfilaments in opposite directions to shorten a fiber and to contract a muscle, but now walking them in the opposite direction to lengthen them, to push them in opposite directions and force the cell to elongate. Okay, and so that's this point here. That motor proteins on these overlapping non-kinetochore microtubules, they're walking in opposite directions while physically attached to both microtubules, and it's pushing those microtubules apart. Okay, and elongating the cell. Okay, because you have to remember that it's not just the kinetochore microtubules that are working, okay, to separate the genetic material. They're directly working to separate the chromatids, but the non-kinetochore microtubules are also working to ensure that the poles are further apart, so that when you do separate the sister chromatids, they remain separated. Okay? Make sense? Questions? Yeah, Levi.
I don't know if you've heard of this already and I missed it, but what's the difference between the kinetochore and a non-kinetochore?
Kinetochore is a microtubule that's associated with the chromosome, and then the non-kinetochore are not associated with the chromosome.
Okay.
They're instead functioning to elongate the cell. Yeah. All right? Nothing? Okay. Moving on. So the next thing we need to talk about, and this is one of the last topics we're going to cover in Chapter 10, our chapter on mitosis, and it's basically what controls does the cell have over this cycle? Because when you learn it and you're just like, okay, the cell cycle is G1, S, G2, all of which are interphase, and then it goes through M, it just gives you this idea that these cells are they're just destined to move through the cell cycle time and time and time again. But we already talked on Wednesday that we know that's not true, because most of the cells in our bodies, they never leave G1. And so because of that, we say they're even in G0, even though they're really just in G1, but they've left the cell cycle. And so we know that these cells have control over where they're at and what's happening during this process. Okay, and so here I want to show you the two different combinations. Oh, this was a really neat experiment that was done, and this is not in your textbook, but it's in the slides, and you have access to this. And so they took cells that were basically stuck in G1. So they took a cell. Let's say you took a random cell, random muscle cell out of one of your skeletal muscles. And then it's basically stuck in G1, or it's in G0. And then what they did is they brought that cell into contact with a cell in S phase, and it induced that cell in G1 to move into S phase and to start duplicating the DNA. So it's like while that cell was keeping itself in G1, something in the external environment now told it to move to S phase. And a similar experiment, let's say again, you take that muscle cell that's locked in G1 and you bring it into contact with a cell that's in M phase, it induces the cell to move all the way through to M phase. But look at this, with not the right amount of DNA. It basically bypassed S phase and G2 and went straight to M phase. You see this? This is normal M phase. The DNA's been duplicated. We've got two sister chromatids. Here it's in M phase, but the DNA was never duplicated. So we're like, there's something chemically that has to be present in these cells to tell them what stage they're in. Okay? There's something chemical. There's something chemical that when present inside of these cells tells the cell what stage it should be in and what should be happening. Yeah, Chris.
What would happen to that one cell that was just like trying to tear that one piece of?
Yeah, I mean, it'll get pulled to one pole or the other. It won't. It can rip them apart, but if the two kinetochore microtubules from opposite poles are attached to pretty much the same place, one of them's going to win that tug of war, and it'll be pulled over, and then ultimately you would generate two nuclei with half as much DNA as they should have. Okay, so I'm going to show you a figure, and initially it's going to be really confusing, okay? But we'll walk through it. We won't run through it. We'll walk through it, okay? So here's the diagram. And so this diagram in the very middle is showing us what? What is it showing us in the very middle of this diagram? The cell cycle. And it's showing us G1, S, and G2, which are all collectively interphase, and then it's showing us M phase. And it's demonstrating what are those chemical components that have to be present to trigger the cell to move from one stage to the next. To trigger the cell to move from one stage to the next. Okay? And here, notice that we have two different chemical components that have to be present in order to make this happen. So we have this enzyme that's called CDK, which stands for cyclin-dependent kinase. But if you just remember it as CDK, that's great. If you remember CDK, I would be incredibly excited. Okay? So here we have CDK. We've got CDK2 binding to two different chemical components in order to get the cell to move from G1 into S phase. Do you see that? So early in G1, CDK2 has to bind what we call the G1S cyclin to start to prepare it for S phase. And what does a cell need to do to prepare for S phase? What happens during S phase? We duplicate the DNA. And what do we need to duplicate the DNA? We learned this in our chapter on DNA replication. What do you need?
You need the DNA material. You need enough material.
You need a whole bunch of nucleotides, DNA nucleotides, and you need all of the molecular machinery you need to duplicate the DNA. You need DNA polymerase. You need primase. You need helicase. You need ligase. You need single-stranded binding proteins. You need topoisomerase, right? all of these molecular machines that we talked about. So in order to even prepare for that, the cell needs to start transcribing the genes that code for those molecular machines. Okay, and so that's basically what gets triggered when CDK2, which is an enzyme, binds to this specific material, the G1S cyclin. It starts to transcribe the RNA that will be translated into these molecular machines. Does that make sense? And then CDK2, the same enzyme, now needs to bind a second chemical. We call this S cyclin, and then that will allow it to actually move into that process and to start duplicating the DNA. And so what happened when we took, in that experiment that I showed you in the previous slide, we took a cell that was locked in G1, brought it into contact with a cell that is in S phase. That cell that was in S phase shared some of that S cyclin with that neighboring cell and induced it to basically say, you're in S phase now. Start duplicating the genome. Which is interesting because it probably wasn't prepared to do so and couldn't do correctly or very efficiently. Okay? And then to go from S phase into G2 or M phase, we need to have a second enzyme, CDK1, bind a different cyclin altogether, the M cyclin. And then now that will allow it to move from the S phase into G2, and then the G2 is basically a preparation for M phase. And what does the cell need in order to execute M phase, in order to go through mitosis? Yeah, Allison.
Needs to rearrange its cytoskeleton.
Needs to start rearranging the cytoskeleton, which will start, you'll start breaking down that cytoskeleton during G2, because by the time prophase starts, you need to start building the spindle. And if you're going to build that mitotic spindle in prophase, you better have the material necessary to do so, right? Which can only happen as you recycle away the old microtubules, okay? We also need to replicate the centrosomes, okay, those microtubule organization centers, okay? And then the cell needs to grow a little bit and just to be able to more efficiently isolate the pieces of DNA apart from each other, okay? And so when you saw a cell that was locked in G1 immediately go into M phase following exposure to a cell in M phase is because the cell in M phase shared this M cyclin with that cell that was in G1, and it immediately told that cell start M phase. Because now it had that M cyclin, and it's already made these CDKs, these cyclin-dependent kinases, they're just waiting to be activated, and now it has the chemical that activates it and moved it into M phase before it even did S phase. Yeah, Chris.
So where do those cyclins come from? So if the CDK...
The cell typically makes them. Yeah, it's a good question. The cell typically makes them. They are proteins, typically, and the cell will make them in response to being told to divide. Yeah, in response to being told to divide. And sometimes the only thing a cell needs to be told to divide is to no longer be physically contacted to any other cell. Because usually if cells, and we'll talk about this in a minute, usually if a cell is physically attached to a neighboring cell, it'll basically tell it not to divide. We call this contact inhibition. And it's because it's usually a signal to the cell that it's a pretty dense area. Yeah. But anyways, we'll talk about that in a minute. Any questions about these? Okay. And so before we start this next question, which is what internal controls does the cell have over the cycle? Because I showed you an experiment where we basically, it was something external that was going into the cell and triggering it to move from one stage to the next. Before we address this question, I want you to do this. I want you to take a couple of minutes and working with those around you, I want you to come up with a list of reasons why cells cannot just keep going through the cell cycle. Okay? A list of reasons why the cells in our body can't just keep going through the cell cycle. Why do the cells need to stay outside of the cell cycle unless there's something present, okay? Or another way to ask this question is, what is the benefit of controlling when you move from G1 to S? When you move from S to G2? When you move from G2 to M? Okay? 2 minutes, starting now.
What's that? Oh yeah. that.
Yeah, it's okay.
Yeah, and there's like a cell.
So,
The there's like you shut it down, there's have chemicals just to make sure that it doesn't go through and reproduce. Or like it's like a dense area, you know, it's another one. So if cells are touching one another, that is contact
that's what
inhibitor, which means that contact inhibition. Or like if like two cells are touching one another, they're gonna be like, oh, okay, like I don't need to reproduce right now because like this is already a dense area, there's already a lot of cells.
Oh, okay.
Reproduce and make it super thick.
Yeah.
And you know, and that would be good if you
and there's no control.
dense cells, cancerous.
Then you're just gonna
And
have
then, you know, there's some cell types that obviously do not have cancer. You know, like
yeah, they're not supposed to do it. So that's probably the reason for all those like checkpoints. Yes, so is to make sure that nothing's screwed up.
Yeah, for sure.
Oh, is that all?
All right. Somebody give me a reason. Somebody give me a reason why cells need to control when they move from one phase to another, when they go through the cell cycle. Levi.
In a more in the multicellular
Mhm.
organism, if you had your cells just constantly reproducing, that would cause just some major problems, like your tissues would grow really fast.
They would.
Like cancer, that's basically what cancer is, right? It's just
Yeah, it doesn't. That's what you call like a tumor. That's what happens when you start to form a tumor. It doesn't become cancerous until something else happens, and that is the cells lose their ability to connect to neighboring cells and to start to spread and start to penetrate other tissues. That's when it shifts from just being a tumor to being cancer. But yes, I mean, ultimately, when you've got cells growing out of control, it'd be very difficult to maintain healthy subpopulations of cells. It'd be very difficult to maintain proper tissue size, tissue placement. Yeah. Allison.
And the cells wouldn't have any time to actually perform their function.
Yeah.
It's constantly replicating.
That's true, because you basically have two. Yeah, I mean, basically you have two phases of the cell cycle. The well, you have M phase and then you have interphase, but you basically have two different varieties. One is grow like crazy, and the other one is do some task. But if you're just in actively mitotic tissues, you're right. I mean, they're basically just growing like crazy. They're not. They have very little time to carry out normal cell functions, whatever you need to happen for that tissue. Good. These are all like for the good of the tissue. For the good of the tissue. Any ideas of why a cell needs to do this for the good of the cell? An individual cell. Yeah.
Well, like you said, the contact inhibition.
Mhm.
So if there's too many of them and not enough room, they wouldn't be able to do their thing.
Absolutely. Well, they'd also risk something else if there are too many cells. What would they risk? Yeah.
So I think the energy part.
Yeah, the energy part, they'd risk starvation, right? You'd risk starvation. Absolutely. Any other ideas for the good of the cell? So a cell's success, if you will, an individual cell, and it's weird to think about it this way in the context of a multicellular organism, but the success of a cell is determined by its ability to replicate itself perfectly, right? The success of a cell is determined by its ability to ensure that when it goes through the replication process, through both M phase and cytokinesis, that it produces daughter cells that are identical to itself. Okay, so this gives the cell some control to ensure that everything is done correctly. That when the DNA is duplicated, it's duplicated correctly. And if it isn't, to go and fix it before it moves through the cell cycle. So this gives the cell a layer of control to ensure that, again, everything is done correctly. So that cell, when it goes through mitosis and cytokinesis, it accomplishes making cells that are identical to itself. Davies.
Is that the reason why we have checkpoints in those?
Yes. And so those checkpoints are looking for two things. So in the previous slide here, let's go back to here. We've got the G1S checkpoint, the G2M checkpoint, and the mitotic spindle checkpoint. We're going to talk about all of these in a little bit more detail in the coming slides. Those checkpoints are looking for two things. One, that you have the proper CDK, that enzyme, bound to the proper cyclin. And the other thing it's checking for is for fidelity. Checking that everything is done correctly. If it moves from G1 to S, it's checking to make sure it made all the machinery right to do S phase. To move from S to G2, it's checking to make sure the DNA was replicated correctly. To move from G2 to M, it's checking to make sure that it has everything it needs in order to do M phase correctly. The M checkpoint, you'll see this in a minute, is ensuring that the kinetochore microtubules are attached to both sister chromatids, so that mitosis is done correctly. Okay, so it's checking for fidelity, and it's also checking to make sure the proper cyclin and cyclin dependent kinase combos are in place. All right. Okay, so what internal controls? A couple of researchers, Robert Johnson and Patu Rao. What they did is they took these special cells called HeLa cells, which are cells that have been grown from a woman who died from cancer in the 1950s, I believe. Her name was Henrietta, what was the last name? Lacks. And so these cells have basically lost the controls on the cell cycle, and they just keep going through the cell cycle. And they are, in one way, they are immortal. And so what they did was they took these cells and they wanted to know what things have to be in place in order for cells to do this, to just keep going through the cell cycle. Keep going through the cell cycle. And they use this to actually determine and find our three main checkpoints. That is our G1S, our G2M, and our M checkpoint. And so this G1S, this happens to be the main checkpoint, which makes sense because if a cell can only function normally in G1, and if once a cell gets to S phase, it's basically committed to finishing the process, that main checkpoint should be at G1S. Okay? And then we've got G2M to make sure you have all the materials that you need in order to progress through M phase, the M checkpoint or mitotic spindle checkpoint to ensure that things are attached correctly. And so the cell will never go to S phase unless the DNA is undamaged and the cell has the right growth factors in place, those chemicals telling it to grow, and it has enough energy necessary to finish the process. Okay? And so if the cell does not have that, it will remain in G1. And you're like, well, if most of the cells in our body are in G1, or if you want to call it G0, that's fine. Does that mean that the DNA is damaged or that they don't have the energy components necessary? No, they don't have the growth factors. They're not being told to grow, to replicate. But see, cancer cells lose their ability to sense growth factors. They're always active. They're basically, it's almost like they're in an environment that's just telling you, keep replicate. Oh, another thing that I forgot to mention where the cell does it for the good of the cell is we talked about this with DNA replication. Every time a cell replicates, the telomeres shorten, right? Every time a cell replicates, your telomeres shorten, and after a certain number of replication events, your telomeres are short enough that you're starting to lose information, valuable information. And you're like, so how do cancer cells then just keep growing like crazy? One, they're not so concerned about losing information. They're just concerned with replicating as fast as possible. And then two, they overexpress our enzyme that helps to prevent rapid shortening of the telomeres. And what was that enzyme again? Do you remember? Telomerase. Do you remember talking about that in DNA replication? Man, it's really unfortunate that DNA replication will not be on exam 2 because I feel like you've lost it. You've lost it. And it should come back because the final exam will be partially cumulative. It's going to be mostly material we cover between exam 2 and the final, but there'll be some things, and you can anticipate DNA replication coming back, making an appearance. Yeah.
So if the cell is deciding whether it's going from G1 to S, is it taking in external factors? Like
Yes, it is.
So does that mean that when a cell is going to replicate, they're all replicating simultaneously? So
Typically so. I mean, maybe not all of them, but many cells are replicating
So
simultaneously.
your body will go through periods where it just remains in the G1 and then if factors change, they all go.
Most of the time it's tissue specific. So usually you have tissues that are actively mitotic because they have to be. And so in the dermis, so right at the bridge between the dermis and the epidermis, you have a tissue that is actively mitotic. It's constantly dividing because you're constantly losing cells from your epidermis.
So like
Right.
if you were to go to the gym and worked out your arm really, really hard, would that stimulate it? Or-
No, so with muscles, it's usually not a matter of generating more cells. It's generating more protein. Yeah, it's the cells starting to express more protein because of the strain that they're under.
And that'll cause your body to grow.
Yeah, to bulk up. Yep, as those muscle cells start to make more of those protein fibers, the individual cells will grow. Yeah, most growth and development is done by cell division. That's what we talked about on Wednesday. But some of the growth and development that happens after you've left that growth and development stage, because there's really two stages of life. There's the growth and development stage, and then there's the pathway towards senescence, right? And so once you leave that growth and development phase, as a mammal at least, reptiles, they never stop growing. They just do growth and development forever. But mammals, once you leave that growth and development stage, any growth that happens after that is usually a matter of increasing cell size. In all aspects, actually. If you're going to bulk up, it's increasing the size of individual muscle cells. If you're going to gain, a little bit of size around the midsection, it's just packing more fats into adipocytes. Although you can make more adipocytes as well, if you need more space, because there's only so much fat you can fit into one adipocyte. So
Going through mitosis would more mean to replace some.
typically, typically, yes. Yep, if you need to replace something or you're at a point in development where you still need to grow. You've got a lot of development left to go and your tissues need to grow. Yeah. Now, bone density can increase as well due to strain, and that's typically not either cell proliferation or cell growth. It's reinforcement of the matrix. Most of the bone density is made up of matrix and not individual cells, and so it's just reinforcement of the matrix. So when you look at somebody that's like a boxer or somebody that's like heavily involved in contact sports, their bone density is greater than somebody who isn't, because their bone cells are basically induced to further reinforce bone density. Yeah.
So, when you're young and you are growing, what is the pain that's called? What's that? Growing pain.
Growing pains.
Yeah. What is?
Yeah, I mean it's just the proliferation of cells is putting pressure on nerves. Is oftentimes what's going on. Is just the proliferation of cells is just it's either creating inflammation or it's more often just putting pressure on nerves. Yeah, that's a good question. Yeah, Davies.
How do brain cells relate to this?
Yeah, brain cells, it is very, very difficult to induce brain cells to go through mitosis. So when you have nerve damage, it's very, very difficult to restore function. Usually what happens more often is one nerve will pick up the slack, and rather than you'll regenerate a damaged nerve. And so that's why usually nerve damage is permanent or at least semi-permanent unless you can get another nerve to pick up the slack. So in some tissues, the nerves are close enough together that you have some redundancy. But in a lot of tissues, if you've got nerve damage, it's a permanent situation. So, and you can see that with people who have nerve damage associated deafness, that it's almost impossible to restore hearing. Because you've got basically a single main nerve that controls those signals, and it's very difficult to get nerves to regrow. Yeah. Now, if you have a significant muscle tear, that's a different thing. You can induce muscle cells to go back through mitosis, and or at least recruit some multipotent stem cells, not pluripotent, but multipotent stem cells to come in and differentiate into muscle cells to replace those that were damaged. But yeah, it's very difficult to do with neurons. Yeah.
There's some incidents where when somebody is cut from a certain side, like they replace that skin using another part of the skin outside.
Yeah, it's just a stop measure to prevent infection. So when you're doing a skin graft, and you do it often with people who are burned, because you want to reduce the probability of infection, and the skin is the primary and the best barrier against infection. And so it's not that skin's going to take. I mean, it's possible that skin could gain a blood supply and survive. Usually it's that skin's just kind of taking the place while the other skin is being regrown.
So
Yeah.
do they remove it or?
You can, yeah. And then now sometimes with burns, that skin will eventually gain a blood supply and will remain viable, and that's why you can see with some people if after burned, if they get really really good care, that you can really reduce scarring, and it's because they might get those skin grafts to actually take and to gain a blood supply, which would minimize the scarring, versus if you just have to regrow that, because you're not going to regrow it normally, and it's going to be mostly scar tissue that ends up taking that place instead of what would be normal skin. Yeah. But that's why, usually when you have people that experience really traumatic burns, if they have the means to do so, they usually will go to hospitals with like the absolute best plastic surgeons, to try to minimize scarring. Okay. So the cell will remain at G2M unless the DNA was copied correctly during S phase and it has what it needs to carry out M phase. And then the mitotic spindle checkpoint, again, this is just, it's assuring that the chromosomes are attached correctly, so that when your kinetochore microtubules start to shorten and your non-kinetochore microtubules are pushing the poles in opposite directions, that the chromosomes separate properly. Okay? That make sense? These 3 checkpoints, we okay? All right. So continuing this question, another group of researchers, they wanted to basically study what genes are responsible for expressing the products that the cell needs to move through those checkpoints. What genes tell the cell to start making those cyclins, or making those cyclin-dependent kinases. And so they found they found a gene called CDC2 that codes for these cyclin-dependent kinases. Okay, CDC2, and these cyclin-dependent kinases, we've already mentioned this, they're activated by specific cyclins, and they usher in moving from one stage to the next. Okay, so again, remember all of those checkpoints, the G1S checkpoint, it's checking to make sure that the cell's ready to go through S phase, but it also needs to check to ensure that CDK2 is bound to that S cyclin. Wow, this is coming out of order. Sorry, I had to bring it all because that one's coming out of order. So these kinases, it's an enzyme, we've seen this term before, it's an enzyme that phosphorylates things. And so these kinases tend to provide the on switch to several other enzymes. And so this on switch happens to be this phosphate. Yeah.
For the kinases, phosphorylates things. So
yes,
does it adds a phosphate group?
It does.
Can it also remove a phosphate group or no?
If it did, it would be called a phosphatase and not a kinase. There's typically going to be a separate enzyme called a phosphatase that will remove the phosphate groups added by these kinases.
Okay.
Because if you phosphorylate some enzymes, it turns them on. The only way to turn them off is to remove that phosphate group, or degrade the entire enzyme, right? And so there are other enzymes called phosphatases that'll remove that phosphate.
Okay. Thank you.
Yep, you are very welcome. So the cell controls this entire process by synthesizing and degrading specific cyclins. So if it wants to move from G1 to S, it makes the G1S cyclin and it makes the S cyclin. So that when they bind to CDK2, the cell is able to go from G1 into S. If the cell wants to go from S into G2, it needs to make the SG2 cyclin that will bind to CDK1, okay, or the M cyclin rather. The M cyclin that will bind to CDK1. All right, so we already mentioned this. G1S cyclin binds CDK2 near the end of G1. But you see this G1 cyclin also binds 2 separate CDKs, CDK4 and CDK6. And so what that means is that first main checkpoint, that G1S checkpoint, requires 3 different kinases to bind 3 different cyclins. It requires CDK2 to bind both the G1S cyclin and to bind the S cyclin afterwards, right? Do you remember that from the chart? It also requires CDK4 to bind G1 cyclin, and it requires CDK6 to bind G1 cyclin. So you have 3 different cyclin-dependent kinases binding 3 different cyclins in order to move from G1 into S. And so it's the main checkpoint and the cell has the most control there. Yeah.
You mentioned on the quiz CDK3. What?
Yep. So the question on the quiz says which CDK is not required, and as far as I know, there is no CDK3.
Oh, really?
Yeah.
At all, ever?
I mean, they're not in this process. There
Okay.
may be a CDK3 involved in something else, but it's not involved in the cell cycle.
Gotcha.
Yeah. Okay. And then this last point just sums up what we talked about through that diagram, that the M cyclin binds CDK1 and allows the cell to move from G2 into M phase. Any questions about these levels of control? So again, the cell invests a lot of time and energy and resources into going through the entire cell cycle. So it needs to ensure that it is completely ready to do so. It's completely ready to do so. All right. Yeah. All right, this next question. How does an organism maintain subpopulations of cells? How does an organism maintain this? Because it seems to be that if you can trigger a cell to do this, how do cells work together to ensure that an individual cell isn't going to just grow like crazy? Because it seems like it would be to the advantage of the cell to just grow like crazy, even in a multicellular organism. So all of these internal controls, the coding of CDKs, the coding of cyclins, the degradation of cyclins, all of those are controlled and modified by hormones, external signals, and by growth factors. And so these hormones and growth factors will trigger processes that will speed up if necessary, slow down if necessary, completely stop if necessary, these internal cellular processes. So it's a way surrounding cells can control what's happening in neighboring cells. Especially cells that are touching each other experience what we call contact inhibition, and they will move into G0, which is really, it's G1, but it's G1 where it is in no way prepared to move into S phase. And then this is what you were asking about, Levi, is losses in the cell cycle control will give rise to uncontrolled cell growth, and it will give rise to tumor formation. And then for that to become cancerous, those cells need to also lose their adhesion to neighboring cells. And so at that point, then they can start to move around the body, and they can be permanently shut down from ever experiencing contact inhibition. And then that way, even if they penetrate a neighboring tissue, they will still keep growing like crazy. And so these shifts from normal cell cycle control to abnormal cell cycle control usually results in mutations in what we call oncogenes, which are the genes that code for the machinery necessary to move from one stage to another. Yeah, Allison.
So do metastatic cells then induce the cells that they metastasize to go through the cycle? Like
No, I mean they can. Like in that experiment that we saw, if you take a cell that's in M phase and you bring it into contact with a cell that's in G1, it can. So it has the possibility to do that. The thing is, these metastatic cells, they don't really communicate with neighboring cells anymore. So it'd be difficult for them to transport material through that. So what happens there is, remember when we talked about our different junction types? One of our junctions, they're called gap junctions, and they connect the cytoplasms of neighboring cells. And so if you take two cells and you bring them together and they're capable of behaving like normal cells, sometimes they'll develop those gap junctions that will connect them, and then will allow, say, M cyclin to freely move from one cell to the neighboring cell, which would then induce the cell to go into M phase, even if it wasn't ready for it. Yeah, Chris?
So for cancer, like isn't there a difference between like a benign, like someone says like, oh, it hasn't metastasized yet, it hasn't spread throughout the bodies, it's cancer but it's benign, is that still cancer if it is benign, if it's not?
No, if it's benign, it has not metastasized. Now, any tumor has the potential to develop into cancer, which is why they're concerned with tumors even when they are benign. And then another thing is they can put pressure on neighboring tissues, right? Even if you have a benign tumor, but it's in your brain, right, it can cause very significant issues. And so you're really concerned about two things. One is inhibiting the function of the surrounding tissue, and then two is that it could lose the connections and then spread. Now, even if it's lost the neighboring connections, but it hasn't gotten into the bloodstream yet, it can still be cancerous if it's penetrating that tissue. And it's not just growing and remaining a roughly spherical tumor, but it's actually penetrating and starting to form like finger-like projections into those neighboring tissues. So you can have cancer that's still localized, that hasn't gotten into the bloodstream and start to spread, first to the lymph nodes and then to other tissues. It can still be cancerous because it can still have lost that contact inhibition and be penetrating that tissue.
Okay.
Yeah. Davies.
For people that can't grow taller, like
Yeah.
Is it that their cells have gone to gene one or
For people that cannot, yeah, I mean, so typically you're either programmed to not develop as many cells, so somebody who's taller, so taller and larger people tend to have more cells than smaller and, shorter people. And so you may not have been induced to grow, and that could be a genetic reason. 80% of the variation in human height is due to genetic differences, but about 20% of it is due to environmental differences. So you could have been programmed to not replicate as much and produce as many cells, or it could be due to environmental reasons, that you were, say, programmed to be a certain height, but you didn't get the nutrition necessary for your cells to go through the cell cycle as quickly as they should have.
is there
Based
a
on your
point
program.
when cells can't replicate anymore?
There is a point where cells can't replicate anymore. Yep.
And
Yep.
where is it?
It depends on the tissue. Yep, it depends on the tissue. Yeah. It's usually somewhere between 50 and 70 replications. Somewhere between 50 and 70 replication events, and then the cell can no longer replicate because the telomeres are too short. And so it'll permanently shut off replication. But then in certain tissues where you need to just actively replicate for your entire life, like in your dermis, epidermis boundary, or in your gametes, or in your gonads where you're making gametes, those tissues express telomerase. And so the chromosomes don't shorten as much. And sometimes they don't shorten at all. Yeah. All right, last question. How does binary fission differ from mitosis and cytokinesis? Simply put, it's way simpler, right? Because you obviously can't have mitosis if you have no nucleus, right? Because mitosis is nuclear replication. Yes? Mitosis is nuclear replication. Cytokinesis is the splitting of one cell into two, and so binary fission is what prokaryotes do. And basically the only things they need to do is they need to replicate the DNA. So they have to do something equivalent to doing S phase in a eukaryotic cell, and then they need to do cytokinesis. But we call this binary fission. And so some of the things that you don't find. So the chromosomes usually don't become visible during binary fission. They don't need to condense like they do in eukaryotes, because they don't need to be as associated with specific. Well, there are microtubules in bacterial cells. We're not really sure how the chromosomes segregate. It's probably that the chromosomes are actually physically attached to the plasma membrane in several places. And so as you start to divide that cell and pull it apart, it just pulls the two chromosomes separate. Most prokaryotes have a single circular DNA, so we don't need to separate sister chromatids. It's basically one chromosome, and as it's being replicated, just naturally separates during that process in the same way chromosomes would in eukaryotes if they don't remain attached together at the centromere. In prokaryotes, DNA replication takes most of the cell cycle. So in E. coli, the organism that we know best, 19 out of the 20 minutes for a replication event, basically every 20 minutes, E. coli, the number of E. coli will double. We call that the doubling time. And so every 20 minutes, every cell will become 2 cells. 19 out of those 20 minutes are devoted just to replicating the chromosome. Just to replicating the chromosome. Yeah, Chris.
Is that just because that's what its function is, just to reproduce like crazy? It doesn't really happen.
Well, sometimes. Yeah, now it doesn't always grow like that. Sometimes it will grow like that. It only grow like that still if those cells have the nutrients necessary to go through that process. But why is it that way? I mean, it's really all it needs to do is replicate the chromosome and then split one cell into 2. There's not a whole lot else that it needs to do. It's not as complicated as going through mitosis. We have one more question to deal with, but we don't have time. So we'll go ahead and call it quits on our day and on our week. Keep in mind the study guide, it's up on Canvas. Engage it. If you have questions, ask them, and we can work through them.